Okay, so to finish out the 3.3a unit, we're going to be talking about ways that we get genetic variation through meiosis. So you'll probably already know that you're not identical to your siblings, even though you share so many genes with them, you're not identical to your parents. So we're going to talk about some of the mechanisms of how that occurs. And we've already touched on some of them, but we're going to be looking at them more in depth right now. So genetic variation is necessary for a species to change and evolve over time. So during meiosis specifically, there are two stages that promote this. And the first one is those crossovers that happen in prophase 1. And the second is the random orientations of the chromosomes that are happening both in metaphase 1 and 2. And remember, metaphase 1 and 2 is where the chromosomes are aligning on the middle. And who's on which side of the cell is random. So we'll look at each of these a little bit more in depth. So let's take a look at the chromosome or the crossovers in prophase 1. So let's assume that these are a pair of homologous chromosomes. And remember, homologous chromosomes have the same set of genes in the same orders, but maybe they have different alleles. So let's say on the green, I have a gene for A and a gene for B and a gene for C in these particular locations. And then let's say for the yellow, I'm still going to have the A, B, and C, but maybe they're different versions of that gene. So in one version, they're going to be these uppercase letters, and in the yellow, it's still gene A, but I'm indicating allele by a lowercase a, lowercase b, lowercase c for each of these alleles. So they each have the same set of genes, but just different alleles. So what's happening during the crossovers in prophase 1 is they're actually reaching out and intertwining. And if you look at pictures of these on the internet, you will actually see the chromosomes reaching out to one another and getting entangled. And what they are about to do is swap sections. So the green one is about to donate its capital A to the yellow, and the yellow is exchanging to the green its lowercase a here. And it happens randomly. For some genes, it might not happen at all. So say for the B alleles, they're not part of this. They're going to stay exactly where they are. But let's say for C, this is crossing over here. Let's say for the C gene, they're going to swap alleles. So what you end up with is one chromosome that is completely as it was before. So the green is maintaining those uppercase versions, those alleles of gene A, where they're uppercase A, B, and C. And you might still have some of these where the yellow, the other homologous chromosome, is maintaining its set of alleles of the lowercase a, b, and c. But the crossovers now created a new set of alleles on a chromosome. So for this one here, it took the lowercase allele from the yellow, it kept its uppercase b, and it swapped its uppercase c for this lowercase one. And so again, for this happened on the other side too, right? So for this chromosome in the center here, it exchanged its section for the uppercase allele on green. It kept its own lowercase b, but it exchanged its lowercase c for the uppercase one on the other side. So now you have these two that are completely new set of alleles. This combination did not exist before until crossovers happened. So this is one way that you get genetic variation through the crossovers that happen between homologous chromosomes in prophase 1. Okay, so this crossover again is occurring in prophase 1, and it's the sections of the chromosomes that are being swapped, and it's these new allele combinations. They weren't here before. So these guys are new allele combinations, and that adds to genetic variety. So the other source of genetic variety during meiosis comes from the orientation of chromosomes in metaphase 1 and 2. So let's say we're in metaphase 1, and you know it's metaphase 1 because I only have one cell, and I've got these two pairs of homologous chromosomes. So which side they're facing is random. In this case, my darker chromosomes are on the bottom and the lighter ones are on the top. But it could easily be that they're flipped. And in this case, I flipped them both, but it might just be one that flipped, right? So maybe in a different cell, the dark 2s are on top and the 1 is on the bottom. And you're going to get this for every single chromosome pair. So that's 2 to the 23 options that you have. So what's going to happen is that These random orientations during metaphase 1 and 2 results in nearly infinite combinations of chromosomes. So even without the crossing over, there's already 2 to the 23rd possibilities that you can have of who ends up on what side, because we have 2 of each chromosome, and who's on one side or the other, it's equal chance. So what this is going to do is during anaphase, when they separate, now you're getting these different haploid cells. So in one possibility, the darker chromosomes were on this side and they stay together, and on this possibility, it's flipped. Like, it could go either way. And again, there's two separate metaphases. So let's say that in this one, it's opposite, that the darker chromosomes are on opposite sides. And on this one, maybe it's this orientation. So you can see how right now in each of these 4, they're all different from one another. And then when we separate them again, now you have these, 8 cells that are all distinct from one another. None of these 2 match. And it's because of, who was on what side. And this can happen for each of the 23 pairs. So now they are all unique. So those random orientations during metaphase 1 and 2 is giving you another source of genetic variety. So the last thing, and this is a little bit outside meiosis, but I will be asking you for this, is it's random which one of these gametes is going to win for both mom and dad. So you're getting variety from 3 sources. One of them, the crossovers from prophase 1. The second is from the random orientation of chromosomes in metaphase 1 and 2. And then the third source is that you are getting chromosomes from mom and dad, and it's random which haploid cell wins. It's random which egg happens to be fertilized, and in particular, it's random which sperm wins that fight. Okay, so for summary, how we generate unique genetic variation. Number 1, crossovers during prophase 1 between those homologous chromosomes. 2, the random orientation of those chromosomes during metaphase 1 and 2. And then 3, the fusion of random gametes from mom and dad. So these are 3 sources of how you would generate unique genetic variation. So from there, we're going to move on to non-disjunction. So we've already sort of touched on this topic a little bit, so last week. So non-disjunction occurs during meiosis, and all non-disjunction is a failure to separate correctly these chromosomes in either anaphase 1 or anaphase 2. It can happen either in the first division of meiosis or in the second. Okay, so when they fail to divide correctly, you're going to end up with an incorrect number in some of these gametes. So some of them might have just 1. Some of them might have an extra 1. So in any case, when it's the wrong number of chromosomes in the end, you would have had a non-disjunction event. So in humans, if they have any number that's different from 46, they had a non-disjunction event somewhere in meiosis. So again, we've talked about this already. We know that Down syndrome is caused by a trisomy in chromosome number 21, but you could easily have a non-disjunction in any of these chromosomes, including the sex chromosome. So for example, this individual could have had a non-disjunction where they only got 1 X chromosome. It does happen. So be aware that it can happen to any chromosome. So these chromosomal disorders are connected to age, particularly of women. So non-disjunction is more frequent the older that somebody gets, particularly these trisomies. So again, this is maternal age here on the bottom, and this is the risk of Down syndrome and live births, and you can see that it increases dramatically after the age of 35. So this is the age of the mother. So as parents get older, the risk of having Down syndrome or other chromosomal disorders increases. So here is a table of ages, and this is the risk of the different trisomies. So these are the only 3 that are survivable. And in the case of trisomy 18 and 13, the babies only live like a week. And trisomy 21 is the longest lived one. So you can see that if you had a baby when you were a teenager, you would only have a 1 in 1,600 chance of having a child with Down syndrome. But by the time mom is 45 to 49, that risk goes up to 1 in 20. Okay, so how do we diagnose these in the first place? So there's two main ways. So the first is through amniocentesis and chorionic villus sampling. So both of these involve obtaining fetal cells somehow before the baby is born in order to create that karyogram and determine the human's karyotype. So two methods of doing that. So in amniocentesis, this is a procedure that's done later in pregnancy when the risk of miscarriage is pretty low. So in this one, a needle is inserted through the abdomen, and I'm sure this is just as awesome as it looks. And that needle is punctured into the amniotic sac where amniotic fluid is withdrawn, and that is the source of the fetal cells for amniocentesis that they can then do the karyogram on. The other is chorionic villus sampling. So chorionic is just like is basically referring to the eggshell of the human. And in this kind of test, the tube is inserted through the cervix, and it's actually removing a part of the placenta called the chorionic villi. And this also contains fetal cells. So this one can be done earlier in pregnancy, but it does carry a higher risk of miscarriage. So people have to make that decision for themselves, whether they want that testing at all, and if so, which version and when. So that concludes our unit on meiosis.